Alright, my name is Sierra Gold, and I'm going to teach you about the Lichen Symbiosis today. So to start out, why do we care about lichen? Well, lichen are an ancient symbiosis, one of the oldest that we know of, and kind of a foundational species in the idea of symbioses in general being discovered and considered. In the fossil record, we can see that we have lichens from up to 415 to 350 million years ago. So they really are an ancient symbiosis. And there's also a vast variety of lichen. We'll talk about that a little bit in the next couple slides, but lichen is not only unique but very diverse, which makes it interesting to study as well. And one of the main reasons that scientists are especially interested in studying lichens is their ability to survive harsh environments. They're found on every continent, including Antarctica, and in all kinds of biomes, from Arctic to desert, and everywhere in between. So they're a really fascinating system, and today we'll go over some of the reasons that they are able to be such an interesting symbiosis. First, let's talk about the partners in the symbiosis. So a lot of times when we talk about symbioses, we're thinking of a host and a symbiont, and lichen is so entangled together that there's kind of different words for it. So rather than a host and a symbiont, we use the words mycobiont and photobiont when we're talking about lichen. So our first partner is the mycobiont. This is the fungus part. So a lichen is a fungus and an alga or a cyanobacteria in a symbiotic relationship working together, kind of enmeshed into almost one organism. And we call the fungus partner, which is the one that you can see with your naked eye that makes up the structure of a lichen, and that we call the mycobiont. There are about 20,000 mycobiont species, so there are lots and lots of fungi that have evolved a lichen lifestyle. And about 70% of those lichens are from the taxa Ascomycota. As far as the photobiont goes, this is the smaller ones that you can only see under a microscope, right? And this is the algae or the cyanobacteria. They're specifically called the photobiont because they perform photosynthesis, which we'll talk about a little bit later. But specifically, then your partner is an algae or cyanobacteria, and there are 50 genera of photobionts. So you also get a pretty wide range of different algae or cyanobacteria that have developed this lifestyle, this symbiosis with a fungus. But 90% of those are from the group Chlorophyta, which is green algae. So a lot of times throughout the rest of this presentation, when we're talking about lichen, we'll use the term algae when talking about the photobiont, but it is about 10% of the time a cyanobacteria rather than an algae that's making up that photobiont part of the lichen. So first things first is the currency. So why are they in this symbiosis? What do they get from one another? And the mycobiont gives the algae the structural support and the protection that it can't get on its own. It has that fungal structure. We call it the thallus, is the body of the mycobiont of the lichen. And within that lichen, kind of enmeshed in its fungal hyphae, these kind of strands that you see in this picture, are the actual algal cells or the photobiont cells. And so it not only provides them that structural support, but also gives them protection. We'll talk a little bit about that later, the different forms of protection that the mycobiont can provide the algae with that it wouldn't get on its own. And then the photobiont gives the fungi the products of photosynthesis. So, the products of photosynthesis are energy, right, and sugars. And so that energy and sugars, the fungus can benefit from via that photobiont. And then together, one of the main things that we'll touch on later is a concept called anhydrobiosis. This is the term we use to talk about surviving desiccation. This is one of the things that's especially unique about lichen, is their ability to survive desiccation. And it's only possible from their symbiosis. Both fungus and photobionts are sometimes able to survive a bit of desiccation on their own, but together they have this incredible unique ability to survive desiccation. So how's it transmitted, right? A lot of times, again, we're talking about a symbiont that's transmitted through contact or through in the egg to the mother, from the mother to the child, things like that. But it's a little bit different in lichen. So there's two ways that the symbiosis is transferred from one generation to the next. The first is asexual reproduction. So this is a form of vertical transmission where you go straight from the lichen to a new lichen. And all the algae, the symbionts, both symbionts are here the whole time. Now, the way that this happens is through fungal propagules that we call Seridia. You can see them in this picture, kind of breaking off. That will break off from the main lichen body, and they are basically pieces of the fungus, the hyphae of the fungus, wrapped around individual cells or a few cells of the photobiont. And so they're already together, and then they can just go land somewhere and start growing a new asexual individual or asexually produced individual. The other way is a little more complicated, and it is a form of sexual reproduction. And in this form, it's a horizontal transmission. So what I mean by that is that there's this body on the fungus. Usually it's called an ascocarp. There's other words for it in other types of fungi. There's a lot of interesting terminology in the fungus world. But essentially, it's fungal spores. So just the regular spores that the fungus would use to reproduce, it reproduces, and it sends out those spores, those sexual spores. And then those fungal spores, whenever they land and they fertilize and those types of things, they need to find a new photobiont somewhere in the environment to form a new lichen. And exactly how they find each other isn't super well known, but we'll talk in a moment a little bit about some of the ways that once they're in proximity of each other, of how they communicate with one another to form that lichen in the first place. So this would be what we call the establishment of the symbiosis when they first come together. So this can happen as a result of sexual reproduction, or just in the first place when you've got the fungus and the algae coming together to form a lichen. This process we call lichenization, for good reason. And it starts with chemical signaling. So both the photobiont and the mycobiont have upregulated genes for signal transduction components. So when they're near each other, we get these upregulated genes, and they can communicate with each other, even though they're different species from completely different kingdoms even. And one of the main things that gets turned on with this chemical signaling are ammonium and ribitol transporters in the fungus. So the fungus is producing different chemicals, phytohormones they call them, and different sugar alcohols, etc. While the photobiont is also releasing these kind of markers, these chemical signals. And two of the main chemical signals that the photobiont produces are a sugar alcohol called ribitol and nitrogen in the form of ammonia or ammonium. And it's interesting because we see in the fungus it specifically upregulates genes for ammonium and ribitol transporters, so the ability to receive those signals being sent from the photobiont. It matches up. I mean, and this is, like I said, an ancient symbiosis, right? So there's been a long time for it to specialize in this way. But it's pretty fascinating to see the ways that they are so specialized to each other. The other main players in this establishment are the lectins that are produced in the fungi. These are parts of the fungi that are able to best recognize and bind to those algal cells. So once the cells are close enough, they're receiving the signals, and the lectins can specifically recognize the algal cells and bring them in. And then there's cyclic peptides in the algae that they're producing that are specifically made to attract the fungus. So together they're able to signal to one another, communicate, and come together to form a lichen. At which point the mycobiont will encase the photobiont, and you'll start to get that full, complete lichen body. So then from there, they continue to develop together. And one of the main things that is important in the lichen symbiosis as it continues to develop and work are what are called polyols. So polyols are an organic metabolite. They're forms of reduced carbon that have multiple hydroxyl groups on them. Another word for polyols are sugar alcohols. And some of those main ones are ribitols and mannidols. Here's the chemical picture for ribitol. And I mentioned that earlier, right? We had ribitals. Manitals are another one that's being produced. And these are things that primarily the algae produces. The fungi, I think, can produce some, but primarily the fungi is converting them into usable forms, whereas the algae is primarily producing them. So they're a really good team in that the algae is producing these and the fungi is good at converting them. And so then they're able to use those polyols for different things. Specifically, growth and development is one of the main things that the polyols are important in. And also there's some question or some interesting research being done into whether or not the polyols are important in desiccation. So we're about to talk about that anhydrobiosis that I mentioned earlier, and it's possible that the polyols are partially helping with this ability to survive desiccation. So for anhydrobiosis, let's start with what it is. So anhydrobiosis is the word that we use to describe what happens when there's not water and the lichen goes into this kind of shutdown state where it stops all metabolism and then it just waits until it's rehydrated in this kind of frozen state where there's no metabolism going on, but it's still alive. And this is really fascinating that it's able to do this. So basically indefinitely until rehydrated in lichens. There's been different studies done that have shown that it's unless if it's kept in a regular okay environment, it is able to just survive basically indefinitely until you rehydrate it, and then it just goes back to metabolizing again. Lichens are especially fascinating in this as well. There's other organisms that do anhydrobiosis that are able to survive desiccation. There's different mosses and bryophytes, and even there's to an extent there's some animals that are able to kind of face this lack of water in different ways and lower their metabolisms and those types of things. But lichens can do it within minutes. Most desiccation tolerant organisms are going to need time for a gradual shift. So as a season changes or something like that, or as there's not rain for a certain amount of days or weeks or months, the organism can slowly adapt to that and start stopping its metabolism in order to go into anhydrobiosis. But lichens can do it within minutes, literally minutes. And it's pretty amazing, and we'll talk about in a moment how they're able to do it so fast. But before we get into that, let's talk a little bit about desiccation and what the problem is so we can look at how lichens solve the problem. So why is desiccation bad? I mean, other than just we know you need water for life, so not having water sounds bad. But one of the biggest problems is that it causes what we call oxidative stress. This is essentially a fancy way of saying that there are too many free radicals, also what we call reactive oxygen species, that can cause major issues. So you have your desiccation, lots of reactive oxygen species are formed, and then it causes direct damage to the DNA, the proteins, the lipids, and can initiate programmed cell death. Bad stuff. Loss of viability, loss of life, essentially. It can be really, really damaging and harmful. There are some desiccation-tolerant organisms that are able to take a little bit of damage and then recover if they get water again soon enough. But lichens are especially unique that they're able to basically just shut it down and bounce back almost to exactly how they were before they started desiccating. And so how do they do that? How do you handle desiccation? The main way is via what are called antioxidants, right? So desiccation is oxidative stress. So antioxidants are specific molecules that can, what they call scavenge free radicals. So essentially they just neutralize those reactive oxygen species. Antioxidants are molecules that are specially designed to be able to donate an electron without becoming unstable themselves, which is exactly what you need to quell the reactive oxygen species, those free radicals, to neutralize them so that they don't cause all these issues. Some examples of antioxidants that we're probably familiar with: vitamin C and vitamin E are two types of antioxidants that we hear about maybe in talking about nutrition and things that are in our foods and those types of things. The scientific names for those, right, are ascorbic acid and tocopherol. Ascorbic acid is an especially big one in a lot of desiccation-tolerant organisms, but interestingly, lichen is lacking in ascorbic acid. It doesn't have much. The main antioxidant in lichen is what's called glutathione. This is the primary antioxidant that they use to combat desiccation. So I mentioned that they're able to do it really fast, right, within minutes. The way that they're able to do this is because almost all of their solutions are what we call constitutive solutions. And this means that in the genome, these genes are always on. They don't have time to induce when suddenly they need to dry out and handle this desiccation. And so they just always have their genes on, which sometimes there's trade-offs, right? There's a lot of reasons that other organisms might have inducible solutions rather than constitutive solutions, ones that need to be triggered by something more directly in order to turn those genes on and get those antioxidants flowing, that type of thing. But there's pros and cons, right? And so even though it may be a little bit costly to always have these genes on, it ends up working out well for lichen because of how well they're able to handle this situation. So those solutions include, first, what we call vitrification. This is one of the first things that lichen do when reacting to oxidative stress. And vitrification is another way that scientists will talk about that is they'll call it glass formation. Essentially, it's a glassification of the cell membranes that causes this protection of the membranes so they don't dry up, are able to kind of get into this rigid state. There are proteins literally called dihydrins and rehydrins, and other ones involved as well. I won't go too deep into that right now because that's not really the focus. Is more an overall talking about the symbiosis, but there are proteins that are involved in dealing with it as well that are also constitutive. And then you have your antioxidants, like I mentioned, one of those big ones, those antioxidants and enzymes that are going to scavenge those free radicals and neutralize them. And scientists think that there's probably other mechanisms. It's a pretty complex process, and so this figure from one of the papers that I was researching in preparing for this presentation talked about how kind of the DNA codes for all of these pieces of the puzzle that together allow them to survive this oxidative stress. But one of the reasons that the symbiosis is particularly amazing is I mentioned that there's other organisms that can survive some desiccation, and those include some fungi and some algae on their own. Both fungus and algae are able to survive a certain level of desiccation, but we see there's some fascinating studies out there that show that the levels, the amount of damage that they take or the amount of time they're able to go before being rehydrated is much different than when they're together as a lichen. As a lichen, they can last basically indefinitely in anhydrobiosis, and when they are rehydrated, they get back to full functioning, basically as if nothing has ever happened. Whereas both fungus and algae usually take time to recover, or they don't fully recover at all during those cycles of dehydration and rehydration. And the thing with this is that algae are really good at providing antioxidants. Algae produce lots of them and are really good at making them, but their problem is that when trying to withstand desiccation and go into anhydrobiosis, they are really sensitive to sunlight. I don't know the exact mechanisms going on behind it, but I know that they can die really easily, get burnt by the sun, basically. And so they're not able to withstand too much desiccation because of that, even though they have all these antioxidants because of their exposure to the sun. And fungus, on the other hand, is not very good at producing antioxidants. It has a few, but not very many. And so it is usually on its own, it's worse than algae at surviving desiccation individually. But when it's with the alga or an algae in a lichen, the fungus gives the algae the photoprotection it needs. So it has things called phenols and flavonoids and other mechanisms that act as basically a sunscreen chemical or surface or those types of things. The sunscreen for the algae so that the algae can produce their antioxidants without being harmed or damaged, and the fungus can benefit from all the antioxidants being produced by the algae. So it's a really perfect way that they solve each other's problems and together kind of become this superorganism that's amazing at surviving anhydrobiosis. And basically rinse and repeat this almost indefinitely. Scientists have been studying and realizing that you can dehydrate and rehydrate lichens almost as many times as you want, or at least theoretically. Like I said, they bounce back to almost as if nothing happened when they are rehydrated. So it's a pretty incredible process, and one of the main ways that lichens are able to survive in such insane environments, such as deserts and things like that, where they go through long periods of time without water, and they're totally fine. And that's because of this ability together as a team, as a symbiosis, to survive this anhydrobiosis. The other little interesting lichen specialized thing that I want to mention today, another cool thing about lichen, is something called kleptosymbiosis. So just for some context here, right? We talked earlier about the establishment of a lichen from a fungus and an algae coming together is to form the thallus, that lichen, is called lichenization. There's also something we call relichenization that's shown in the second part of this figure, which is where the two partners could separate and then return to one another, and that is a situation that could occur. And then we have what they call translichenization. This is where kleptosymbiosis comes into play. So translichenization is when you have a lichen that steals photobionts from a different lichen. It acquires those photobionts from the host lichen. So let's talk about what I mean by that. First, the types of lichens that do kleptosymbiosis are what we call lichenicolous lichens. Lichenicolous lichens grow on other lichens. So it's a lichen that's growing on other lichens, and to a bit of sort of a parasitism in some ways, but in some ways not. There's about 257 species of the thousands of species of lichens in the world. About 257 of those are lichenicolous. Often, lichenicolous lichens start off as a fungal propagule from a sexually reproducing microbiont. So there's a lichen somewhere that produces these fungal spores, and these propagules land somewhere nearby another lichen, but there's not necessarily any photobionts around for them to immediately combine with. And so you'll see these lichens growing on top of the other lichens and gaining their photobionts from the ones they're on top of. This is really common in dry locations, which to me makes sense, first of all, because we think of algae and we usually think of wet environments. And second of all, in dry locations, you're going to have those fungi already growing as lichens in different areas, and so just by landing on a different lichen to get your algae, then you don't have to be seeking out somewhere wet where there might be some algae. There's varying degrees of how much this is parasitism or not. Some lichenicolous lichens that perform kleptosymbiosis is truly a klepto, a stealing, where they basically take over the lichen. They take enough of the photobionts that it sometimes kills the other lichen, the host lichen that they're parasitizing. But there are some that only take a few. They basically only take as many algal cells as they need. So they'll take out a few, and then those photobiont cells will divide and grow, and the lichen will begin to be this full lichen again, and it stops taking photobionts from the original lichen, and they're both able to be totally fine. There's even some debate about if this is even in the cases where they harm the other lichen or to the detriment of the host lichen, that this isn't necessarily parasitism, because some people define parasitism as a form of feeding, and technically speaking, the lichenicolous lichen is still making its own energy with the photobionts that it stole from the other lichen. So it's not actually feeding on the lichen that it's stealing from. So there's a little bit of debate, but it is in many ways a form of parasitism that some of these incredible lichens do. So the last thing to talk about is dysbiosis. So dysbiosis, the term we used to talk about when things are out of balance in a symbiosis. But lichen really don't experience much dysbiosis. They're not a delicate system. They can survive temperatures up to 60 degrees Celsius and as low as negative 196 degrees Celsius. This is when desiccated. When they're hydrated, they can still survive pretty intense into the 30s of Celsius heat and pretty intense cold as well. But specifically those numbers is a range when desiccated. They can survive exposure to heavy metals. Some of the research has shown that when they're in locations with a lot of pollution in the air, especially heavy metal pollution, the lichens do just fine. They can obviously survive extreme shifts in water availability, as we've been talking about. They don't need soil. Scientists off for a long time thought maybe lichen were a type of plant, and many plants we know need soil. Not all of them, but many of them. But lichens, because of their unique fungus-algae relationship, they don't need soil. They don't need any nutrients from the soil. They get all the nutrients they need from the atmosphere and from making it themselves. And lichens are so incredible that they've even survived in space. They can survive the vacuum of space and radiation of space, both in labs and literally in space. This here is a picture of a scientific apparatus that was put into space on the International Space Station that had lichen in these little compartments here, and they saw that lichen again was able to just survive it totally fine and keep living its lichen life. So they're pretty incredible, and it's really hard. My point in this is that it's really hard to disrupt their symbiosis in any way. Essentially, if they're too stressed, they just die. So in some symbioses, if there's an imbalance or a stress that happens, the symbionts might leave the host. There might be this separation. But lichen are so stress tolerant that, or are so co-evolved, so integrated in one another, that they never separate. Really, the only forms of separation are when in sexual reproduction or when your algae is stolen, which even then is not a long-term form of separation. And really, they're almost one organism. For so long, scientists thought they were one organism. And they have such a long coevolutionary history that they've really gotten to such a specialized point that there's no situations where the photobionts and the mycobiont leave one another. If there's too much stress, they just die. But that's hard to accomplish, as you can see. So in summary, lichen are a symbiosis between a mycobiont and a photobiont. Their currency is protection for the photobiont and photosynthesis products for the mycobiont. They experience both horizontal and vertical transmission through asexual or sexual reproduction. Chemical signaling is what enables that lichenization. When they first recognize each other, they have specific genes and specific chemicals that are programmed to be able to communicate with one another to form that lichen in the first place. Polyols are an important molecule that is produced and transformed inside of lichens. Those are important for development and perhaps also for desiccation. And as we said, one of their main incredible adaptations is their ability to survive desiccation through extended anhydrobiosis. We also discussed kleptosymbiosis, that there are some lichenicolous lichens that will steal their photobionts from other lichens when forming a lichen. And we talked about dysbiosis, or in the case of lichen, the lack thereof. That's my presentation on lichens. Here are my sources, and I hope you enjoyed it.